to try to do is organize around lecture 11 first, and then we'll, any concepts or things that you want to verify, we'll go through lecture 11. Then if you have any general questions about lecture 11, I'll take any of those. And then we'll get to lecture 12, which I think was the one that we just had, right? Okay. But it's really lecture 13 that's the killer.
Stop it.
But I'm really not kidding you. Like the vast majority of it. Oh, nice. It's going over YouTube. Nice. So I think the next one is really molecular heavy, and then it kind of just changes a little. So we'll see. Do you know if the feed is actually going through?
Not yet.
Not working for you. Oh no. So before it actually gets there then, and we'll figure out if it does get there or not. Do you guys have lots of questions on lecture? Aha! Max is back. You guys have like what kinds of...
It is going, so you're fine. If you YouTube search CJH332, it'll come up as one of the links will be live. Yeah, I think it's still live. Yeah.
But all that stuff I've just been saying, that'll all be deleted, right? Okay, so I guess we'll just start from lecture 11. So last Wednesday, the first lecture that you actually need to know from for the final exam. Do you guys have any specific concept questions, like things that you were not clear about in terms of concepts? So, sorry, your name is? Tasneem, you're nodding your head, so what is it? What's your concept? Homosynaptic LTP?
Not the
Associative, but you're talking about just LTP,
Just LTP,
like the
yeah.
classic LTP.
Yes.
Okay. Is anyone else having problems with homosynaptic LTP? And could I ask, what do you not understand about it?
Like basically how it works, why it's different from associated.
Okay. Okay. So on any given neuron that we would have here, so this would be the axon, this is the cell body, and then we have a number of different synapses that pass by, okay, from a number of, like this is neuron 2, we might have one from neuron 3, and neuron 3 would maybe make synapses over here, okay? So there are a number of different synapses, like literally on this one neuron, there might be 1,000 to like 10,000 different synapses that we see. So right now we see 4. Okay, so if we're looking at, and we'll make it simpler for ourselves, we'll just look at this, and we'll assume that that doesn't make one. So let's look at this. So this neuron 2 has been firing action potentials in a specific pattern, and it's making a synaptic contact here. So this is neuron 2, the presynaptic element for neuron 2. And this is the synapse that we're seeing here that we're interested in. We'll just call this Tasneem.
Yeah.
Yeah.
Awesome.
Okay, so we'll call this Tasneem's postsynaptic node. So what happens is I have a stimulating electrode over here, then I have a recording electrode over here. I want to measure the changes. If it is going to change, I don't know, maybe it will. I want to measure the changes over here. So is that part okay so far? So this is just the organization of homosynaptic LTP. Okay, so what we do, if I can then take just this, and I just blow that up, and if you're okay with how that's organized, we're only looking at this one synapse that's being strengthened here. Okay, so we'll go here. Hello. How are you guys able to open that door? It's magic. Um, so I stimulate here, so this is my stimulating electrode, and you should always have at least one stimulating electrode, and then you have another electrode that records. So if I am doing nothing else, I'm recording from Tasneem's postsynaptic neuron, I'll get this kind of response. You'll learn more about this tomorrow, but this is what we see sort of normally. We're not doing anything yet. This is our normal response, okay? So let's say I fire one action potential through this stimulating electrode. It releases glutamate, and then it binds to the appropriate receptors here, and then I measure from this again. What should I see? So you really only have three options that are logical, right? So it's either going to stay the same size, it's going to get bigger, or it's going to get smaller. Which one will it be? So I fire one action potential. This is what it was before. Now I fire one action potential. It binds and it causes a response here. What will I see? Yep, so it stays exactly the same size because there's no strengthening, there's no trains of activity. So let's say now, so that's what we see under normal circumstances. So let's say I take that stimulating electrode. At this point now, you should know a number of different ways in which I can potentially strengthen this synapse. So I know it's not all part of lecture 11, but it's part of what we learned yesterday. But based on what you learned from lecture 11, what's one way that I can actually potentially strengthen this synapse?
By training.
Sure, so what do we call that?
Oh, yeah.
So we have, you can either call it like high frequency, hello, high frequency stimulation. This is also called tetanic stimulation. And so what happens is I deliver enough of a charge here that I will be able to deliver trains of action potentials just like you said. So I do that. I wait after I've done these three trains of action potentials, and then I wait and I go back and measure this again. If there's been no change at all, I should see exactly the same response, right? But what usually happens on this postsynaptic cell for homosynaptic LTP, because I've been strengthening this synapse, and I'm only concerned about this synapse, therefore it's homosynaptic, it's one synapse, right? So what ends up happening here is that after those trains of activity, now if I measure the response, it gets bigger. So I haven't, I've stimulated this, then I go back and measure this, and I get a much bigger response. That's homosynaptic LTP. That is probably due to one of two different mechanisms that kind of goes to lecture 12, which is the reason why I get a bigger response now for exact. So one thing I forgot to tell you, in order for me to see that, I still have to fire one action potential. It has to release glutamate. Then I see this response here, right? So this is my high frequency stimulation. I don't record right away. I fire one test pulse, and that test pulse is whether I see a difference or not.
Okay.
So that might be the missing part for you.
Yeah.
The reason why this is bigger now is because most likely what has happened, we have the insertion of more AMPA receptors, or what's the other possibility? There's one other one that we talked about yesterday. Sorry? Yeah, the AMPA receptors actually could have been phosphorylated, so they stay open longer. So either of those, either having more receptors to bind to glutamate, or if they bind to glutamate, they stay open longer, means that this cell depolarizes more than it did before.
Right.
So there's a bigger current due to glutamate.
Oh, okay.
So that's the easy case. So that's homosynaptic LTP. Do you guys have any questions about this or anything that I can clear up for you now? Homosynaptic LTP? Yes, hello?
So, sorry, those trains of action potentials would be considered tetanic stimulation? Because I thought tetanic was like not real stop stimulation. But for tetanic, like you're
so-
taking a pause.
So I'm using what it looks like here. I would be probably looking at theta burst or maybe even prime burst if I had 100 action potentials, then that would probably be more similar to Titanic. But it's really sustained trains of action potentials. Any other questions? Yeah, Tina.
How is this different from associative?
Associative LTP is a little different though. So associative LTP, this is again for one synapse.
Yeah.
So if we go all the way back, and for associative LTP, if we go back to that big picture diagram, and we have one neuron, and here's the axon. These are all dendrites. What we've been looking at is from neuron 2, we have been making that one synapse. That one synapse has gotten stronger. We've been using high frequency stimulation to make this one synapse stronger. But let's say, for example, this neuron also made synaptic connections over this way. And let's say, or let's make it even easier. So let's make it like neuron 3. So here's this one neuron, it's been firing, and this has been getting stronger and stronger. That's the easy part, homosynaptic LTP, right? And it makes sense. The more that this is being used, the more glutamate's being released, the more AMPA receptors are tuning up, and at that one synapse, it makes sense that it gets stronger. So what happens to these synapses? So this one synapse over here has gotten stronger and stronger. Has anything happened to these? No, because neuron 3 actually has been silent up until now. It hasn't been firing. Or what else happens is if I'm firing trains of action potentials here and it's strengthening here, neuron 3 hasn't been firing, or neuron 2 may just be firing like one action potential, and neuron 3 is only firing one action potential. That's not a train of activity. There's not enough stimulation to make any of these synapses from neuron 3 stronger. Not yet. But let's say somehow, because there are lots of synapses, let's say somehow we have these trains of action potentials going down neuron 2, making this stronger, and neuron 3 has decided now that while this is all occurring, that it's also going to fire at least one action potential. By our old definition for homosynaptic LTP, this is not a train of activity, right? But what ends up happening is, because this is being strengthened, at the same time, these synapses here will also get stronger as well. This is associative forms of LTP. So the activity and the changes occurring here are driving the increase or strengthening of responses at these other synapses. It kind of breaks the rules that we've learned classically, that we need trains of activity to get strengthening, which makes sense. But here, it's just kind of going along for the ride. As long as this is active and it's depolarizing this cell enough, that depolarization here is enough to drive the activity and activation of NMDA receptors at a distance over here. So that one firing of an action potential fortuitously at the same time means that you get strengthening there. Okay, any other questions? So this is all still lecture 11. So right now in lecture 11 we're going through concept review. So we've gone through homosynaptic and associative forms of LTP. And do you have any other concepts that you wanted to go back over in lecture 11, so last Wednesday? Do you have any, while we're waiting, do you have any general questions about lecture 11? Are you guys all waiting for lecture 12 then? No? Yes, kind of? Okay. Do you have anything else for lecture 11? Anything? Like even if it's just to confirm something or anything else? Yes. Yeah. So, sorry, fast. So, okay. So if we're looking at the short-term changes in plasticity, so this goes like right back to the beginning of that lecture, right? So we have what is known as short-term plasticity, so short-term changes, and then we have long-term changes, which is something that we've been focusing on last Wednesday, this past Monday, and then we'll finish up tomorrow. So one of the things that happens in short-term plasticity, when we drive the synapse, we get very, very brief changes in responsiveness in the postsynaptic cell. So we're still measuring postsynaptically, but those short-term changes that we see are actually very, very short. So it's on the order of like milliseconds or hundreds of milliseconds to a few seconds, right? So that's why it's short term. And one of the things that all of these seem to rely on, even though we're measuring from the postsynaptic cell, these things are usually dependent on calcium, so the presynaptic calcium, and this is also dependent on the neurotransmitter release. So these are short term. It's usually because we have so much calcium left over presynaptically that we get more vesicle fusion, right? So I don't know if you still remember all the presynaptic machinery and synaptotagmin and all those other calcium sensors. There's a lot of calcium, so you get more vesicular fusion and release, so you get a bigger sort of amount of glutamate. That's in the short term. One of the things that happens a little later, though, as a short-term form of plasticity is that if that were to occur long enough, right, that you would actually see, for a very short term, you would see what is known as short-term depression. So there's just not enough neurotransmitter packed into vesicles that even if there's a lot of calcium, there's just nothing to release. There's nothing left in the vesicle. And so as a result, there's no neurotransmitter, and that's where you get the reduction in the postsynaptic response. So there is that short-term phase. So it's just like if you're running all the time, and then you were asked to run after you've sort of stopped and caught your breath a little, and then you're asked to run again. It's hard, right? Because you've been sort of depleted of your energy, so you have to recover a little bit. The neuron hasn't had a chance to do that yet. Again, it gets complicated because an intermediate form of this, do you remember what that is? So this is short term, and then there's something that can last like a minute or two. So PTP, the post-tetanic potentiation, and we don't know that mechanism. It's probably related to neurotransmitters, but we're not 100% sure. That's still sort of an in-the-dark type of mechanism. So this is in contrast to the long-term forms, which is what we sort of have been talking about. We still need calcium, but it's not presynaptic, right? So when we look at it, we're always measuring here. This is what we want to see. Calcium has been dominating in short-term forms of plasticity here. It's causing more neurotransmitter release in the short term. Here, for long-term potentiation, the role is reversed. We need calcium, but calcium now plays a role postsynaptically. For us to see both LTD and LTP, we need postsynaptic calcium, right? And these are lasting for minutes to days or hours to days or longer. So, I don't know if that answers your question. Any other questions from lecture 11? Yes? Why is it a coincident detector? So what is the neurotransmitter that NMDA receptors bind? So it binds glutamate. What do AMPA receptors bind? So they're coincident detectors because at the same synapse, we have two different glutamate receptors. They're both detecting glutamate, but only one is capable of responding, that's AMPA. the NMDA can only respond if it's coincident to AMPA receptors. So we talked about silent synapses, right? So we need that, why do we need the AMPA receptors? Like why are they crucial for NMDA receptor activity? And, right, yeah, and unfortunately tomorrow the very first thing you guys will see when you come back is we're going to revisit this briefly to make sure that you understand that, and then why I asked you to memorize all those different types of pharmacological inhibitors. So if you were to guess, because you'll see it tomorrow, if I were to take that, remember this is like what we were looking at, a change in this postsynaptic cell, and then after LTP it gets bigger, so we were talking about that briefly. If I were to actually draw out the IV curve for what's happening here, what would it look like? Because you'll see it tomorrow and it becomes important. So if you were to take a look at it, the AMPA response would look like this. So if you go all the way back to what AMPA is doing, it actually allows sodium inward and potassium is leaving. And what we'll do is we'll look at this curve again tomorrow. It'll make more sense to you tomorrow. And at the same time that this is going on, the NMDA receptors IV curve, do you think it looks the same or not the same? So at these negative resting membrane potentials, is it open or closed? Closed. So this is like, remember this is more negative, this is more positive. So it's closed, it actually opens, and then it kind of looks like that. So this gets back into understanding what's actually happening at this synapse. So you'll hear about that tomorrow. I know you don't like being scared, Tina, but tomorrow is a really important lecture. There's a lot of material in tomorrow's lecture. Okay. Anything else I can answer for you? I think there's someone else's hand up. Hasn't even, you had the same question. Did you get the same answer? Okay. Any other questions? Really? Yes. Okay, we will get to lecture 12. Are there any other general questions from lecture 11 that I can answer for you now? Any other questions about any figures that you are not sure about? Any other last sort of, I have no idea what it means? No? So are we good with lecture 11? You don't know yet. Are you confident that you know all the material from lecture 11? You don't know yet. Okay. Okay, so let's move to lecture 12 then. Actually, does anyone have a hard copy of lecture 11? Can I borrow your lecture 11? Do you guys sort of remember everything that we talked about in lecture 11 then after this review? And do you remember most of what you learned yesterday in lecture 12? Sort of, okay. Just wanted to make sure. And did you guys learn all of these different AMPA receptor inhibitors and things? You did not? Did we not cover them?
We did, but we haven't had the chance to study them.
Okay, fair enough. Okay, so let me ask you this, because this is the type of question that I would ask. So, so a scientist is doing recording from a hippocampal neuron, and they find that after high frequency stimulation, that when he examines the postsynaptic response, he sees no increase in the postsynaptic current. So what are the possibilities for why he sees no increase? So A, so the neuron contains CAM kinase 2. And NMDA receptors only. Oops. Or the extracellular solution So if I were to ask you this, so you've just done high frequency stimulation, you now go back, you look at the postsynaptic cell, the cell does not show any sign of an increase in the responsiveness. So what am I telling you? Is there LTP or no LTP? No. There's no LTP. So it's high frequency stimulation. So what would account for that? Which one of these statements might be able to account for that? Any ideas? How many of you would vote for A? The neuron contains CAM kinase 2. Neuron only has NMDA receptors, and the extracellular solution contains potassium, or can't tell. So most of you are thinking about this. So I'm not going to just be asking you about what's in the synapse or what sorts of things were the bullet points. I need you to be able to apply what you know. So if I've created a new phosphatase, a brand new phosphatase, and I stimulate the neuron, and that phosphatase is there, and it works like most phosphatases, what might I see in my neuron? So I'm activating a phosphatase, and I'm not telling you any other details other than I'm stimulating the presynaptic cell. What might I see in my postsynaptic neuron? So I might see, for example, post-tetanic potentiation. Would I see, for example, LTD? Would I see LTP? Or can't tell. So this just means that you understand what the role of a phosphatase is and what is related to the phosphatase. Which one would you say? So I'm hoping most of you would sort of pick D, which would be LTP. LTP and phosphatases are kind of correlated with each other, so keep those general ideas and principles in mind. Any questions? So those are not the trickiest type of questions. You'll get slightly harder ones on the final, but ones that you'll have to apply your knowledge. But as long as you know it, you'll be okay.
Did you say LTD?
Yeah. Not LTP. That would be more like kinase related, or inhibiting a phosphatase might trigger LTP. Anything else? So I guess we're moving to lecture 12 then? Yes? Who has questions on lecture 12? General concepts first, then we'll go back to, or concepts first, and then we'll go to general questions. Someone had a question about lecture 12. Okay, so any concept questions on lecture 12? Yes. Okay. Does everyone have questions related to spike timing dependent plasticity? Yeah? So if again, I need to have two cells that are connected together. So I have the presynaptic neuron, and then I have the postsynaptic cell. So the reason why we're using something called spike timing dependent plasticity is if you were to go back to the original definition that Donald Hebb had, the two neurons actually have to be active together. And that's what he originally postulated, even though we kind of subvert that and do trains of activity. The postsynaptic neuron actually isn't being active at the same time. So one of the things that spike timing dependent plasticity shows is that if I have a presynaptic neuron and I fire action potentials, usually they're at 1 hertz, so 1 action potential per second. So this is somewhat more physiological. And if I also depolarize this cell, the postsynaptic cell, so I increase the, or I change the membrane potential and it depolarizes, what do you think happens to the NMDA receptor on this cell?
Is that the same time?
Yeah, so just in general, if I depolarize this cell here and it contains NMDA receptors, what might happen to that NMDA receptor? Any ideas? So you might help it become activated because when you depolarize it, that magnesium might start to leave. You still need glutamate. That glutamate hasn't arrived yet. So imagine for a second that you're depolarizing this cell. So you're activating the NMDA receptor and you're also causing the presynaptic release of glutamate. In theory, would that actually give you the conditions that might lead to the strengthening of this synapse? Potentially, right? So what ends up happening is I generate a presynaptic action potential here, and I depolarize this neuron. If I do them close enough in time or simultaneously, under the right conditions, and don't ask me what happens at 0 because it gets really complicated, but here we're seeing an increase in strength, increase in synaptic strength, and below this is a decrease in synaptic strength. What we see here is if I fire an action potential here and I depolarize at the same time, and then I were to go back and measure what happens, this synapse gets stronger, okay? If I wait, and time is over here, so if I wait a little bit longer, so I depolarize, and instead of doing it right away, I wait like 5 milliseconds or 10 milliseconds, then what I'll see is that the synapse gets stronger, but it's not quite as strong. It's a long-lasting form of plasticity though, so this is just showing you the relative strength. It will stay at that level for like hours, okay? Have I lost anyone yet? No? If you have any questions, today is the time to ask. So what this is showing you is that there is a timing dependency. The spiking just means that it's the action potential. We call an action potential a spike. And so as we go up farther, so we fire an action potential and then we depolarize. We've waited a little bit longer before depolarizing. That synaptic strength increase is getting smaller and smaller. So we might have, for example, this is our normal response. And then here, this might be this big. At this time point, it might be only this big. Here, it's going down even further. And if we went out far enough, so we fired an action potential, 1 hertz, so 1 action potential per second, and then we depolarized all the way out here, we might actually see almost no strengthening. There's just the time window has passed by. They're not correlated in terms of activity. So it fires glutamate, but the glutamate actually isn't enough to open up the enough AMPA receptors to cause enough depolarization to cause this cell to undergo LTP. Does that make sense? So let me flip things around. So what happens if, for example, we depolarize this first and then we fire an action potential? Then things get really interesting, okay? Because you have to go back to that diagram, and I didn't spend a lot of time on it. Is LTD NMDA receptor dependent?
No.
I don't know. How many of you think that LTD is NMDA receptor dependent? It requires NMDA receptors. Okay, so let's think about that from first principles. If I'm depolarizing this, do I potentially have the capacity to activate NMDA receptors? I do. So the NMDA receptors, when I'm depolarizing this first, actually could activate the NMDA receptors. There are some technical things that we have to do to make this work the way that it does here, but I'm not going to get into those technical considerations. It's a little bit different in this particular case, but let me just take you through the scenario that we get here. So under the right conditions that we're not going to get into, if I depolarize this first, and then I fire an action potential. So this is depolarized, magnesium has come off, NMDA receptors are activated, NMDA receptors are ready to go, and then I get a little cloud of glutamate. It might actually cause this synapse to become weaker. And again, we won't go into what the differences are here. This is just something that you'll have to take for granted. If you take the 432 course with Dr. Wooden, you can ask her like the exact mechanisms that allow you to flip these around. So if we continue to do this and we depolarize this first, then we fire an action potential here, but we've waited a little bit of time in the other way, then one of the things that will happen is the LTD that we get down here actually becomes weaker and weaker. It is also time dependent. Okay, so the NMDA receptor only gets primed for a certain length of time, and LTP up here is also time dependent as well, which is why we have that time dependency. Okay, so I don't know if that answered your question. Does it sort of answer your question? Yeah? Do you have any other questions about this or anything else from lecture 12? Nicolette.
So the
Yeah.
are just low frequency stimulation, or?
It is. So technically speaking, and I didn't go into this, and maybe tomorrow I will add that to your repertoire of things. 1 hertz stimulation is generally considered like low frequency. And here, what you're seeing is a low frequency stimulation is enough to cause LTP under the right conditions. You have to depolarize, and you have to do it in the right order, but it can. To make things worse, and tomorrow you'll find out, because I'll add this for you guys, 1 hertz stimulation just by itself causes homosynaptic LTD. So when you have 100 action potentials per second, it usually causes LTP. If you have 1 hertz to 10 hertz, it usually causes LTD, or a weakening. But again, we'll talk about that tomorrow. Any other questions? Yes, you had a question.
Is that the line of best fit?
Yes.
So
Yeah.
how come it does at a certain time points give you a lower stimulation if you do a shorter distance than it does other times?
Sorry, I'm not sure I follow.
When you stimulate it after a certain period of time,
Yeah.
if it's shorter, you should get a higher kind of response, right?
Yeah.
But on the graph, it shows that sometimes you don't get that high response.
Nope, you don't always, but this is like a best fit curve. That's correct. Tina? Okay. Yes. Yeah. Yeah. Yes. Depolarize first and then fire the action potential afterwards. Kind of. The order though is really important. Like pre-firing and then depolarizing. So simultaneously or pre and then postsynaptic activation for LTP. Yeah, Lexi.
So sort of how many action potentials would have to fire in the pre-synapse?
It's 1 hertz
So it's 1 action potential
per second, yeah.
How long would you do that?
Probably do it for about 5 seconds. So about 5 action potentials. You might do it for 10. Depends on like the preparation, whether it's a slice or whatever it is.
So if the time in between the two of them
Yep.
is short enough, and I'm talking about depolarizing and then firing an action potential,
Yep.
that causes a huge amount of LTD.
It can, yes.
And then if it's long, then it won't, right?
No, so there is like a time dependency for both LTP and LTD. After that time window is passed, you can't generate any strengthening or weakening.
Can we talk about what happens with zero now?
No. It gets really complicated. You can do that in your 4th year classes. So if you want to take membrane physiology with Dr. Feng, you can ask her, or if you want to take 432 with Dr. Wooden, you can ask her about what happens at the asymptotes at this particular STDP model. Yes?
I'm sorry, so just when you're depolarizing the postsynaptic cell, are you actually measuring the action potential there or just the
Nope, depolarizing. EPSC is not technically correct because you don't want to. Yeah, you want to measure an EPSC. Anything else? Yeah, Luke.
So I get it for doing this as a model, but in like an actual like rodent or in a human brain, how would they depolarize the second neuron before the action potential is starting to
It's more about being in sync, right? So if there's a group of neurons that are firing and the other group and the neuron is already depolarized, it may respond in a very different way. So we know that happens, for example, in the cerebellum, we get this type of activity. So it really depends on when the activity occurs and how the other neuron has been depolarized. Yes, hello.
For the theta burst stimulation, is
Yep.
it like the same thing as prime burst stimulation?
Yeah, more or less. Yes, there's some slight differences. I just sort of mentioned it in case you happen to run across it in a psychology course. They often will talk about primed burst models, which are very similar to theta burst. Anything else? Any other general questions? Yes.
my question is regarding the in vivo study.
Okay.
I was wondering if, you know how you explained that they take the embryos
Yep.
and that's where they inject the,
It's not an injection.
oh, sorry, they do
So,
that.
so they do inject, but it's not like where you inject directly through the electrodes. You inject in the DNA into an area in the brain, and then the electrodes drive it through the current that's between the two electrodes into a specific area of the brain.
I was wondering if it's relating the fact that I think in the beginning of the lecture you mentioned silent synapses.
Okay.
Does it relate to like do rodents also have silent synapses?
They do. They were found first in rodents, but that's not why we were talking about injecting in like the superecliptic florin and the electroporation model. So we needed to look at GLUA1, which is a subunit of AMPA. AMPA receptors are the things that move into and out of the membrane. NMDA receptors seem to be locked in place. They don't seem to move. It's one of those very strange sort of phenomenon that we don't 100% understand yet, but it's a very interesting area of research too. So AMPAs move, therefore we want to see if we put in AMPA in an in vivo model as it is that pathway is continually being stimulated, do we actually see the strengthening of that synapse in that sensory pathway? And it turns out that we can see that. Any other questions? Yes.
Isn't that causing some kind of activity that can cause phosphorylation?
Yep. Yep. Yep.
But it also means it's often synapse to begin with to cause the cascade.
To cause the cascade. What do you mean?
Like to cause the phosphorylation.
So how do you get calcium entry? Is that what you're asking in the first place? Okay. I'm not sure if I totally understand your question. How do you depolarize a cell that allows NMDA to be activated to cause calcium to come in to potentially activate CAM kinase 2 to then cause insertion of any AMPA receptor? Is that what you're asking? So let me try to address that for you. And this goes into a developmental model that is not on your test. You don't have to worry about it. But if you go back far enough during development, so like the early period of postnatal development, before we have AMPA receptors, we have NMDA receptor synapses only, right? So this synapse only has NMDA receptors, but has no AMPA. And you're asking, how can that synapse be converted to a functional synapse when the only way we can get calcium in is to activate the NMDA receptor, right? So there might be a ton of AMPA receptors here, but they're not ready to go. Like they need that CAM kinase 2. How could I depolarize that? I could, yes, possible, and that definitely occurs. But there's something else that happens in early development. It's like one of the classic things in neuro, we haven't covered it, but it's pretty standard, yes?
Was it how like GABA was more excitatory than inhibitory?
Yes, so during early development, one of the things that happens is we also have at this, not quite here, because this is the synapse. Here on the neck of these types of synapses, we have GABA receptors. So GABA, in early development, when it gets activated, and there are a lot of different studies to show this, it will actually cause the efflux of chloride. Chloride leaves when GABA binds. This is enough to cause membrane depolarization, and it allows for the NMDA receptor to be activated. So if there happens to be enough glutamate around, it will then cause NMDA to have the influx of calcium. But that goes like in a different direction than you were kind of thinking, I think. Any other questions? Is there any other sort of general questions that you have?
So that GFP marker that we were talking about, that was responsive to pH.
Yep. It's fluorine. You need to know it.
Huh?
You need to know it. It's
SEP?
yes. Yes.
Okay, so it showed different colors when it was intracellular versus extracellular.
Yeah.
Isn't the pH of a cell like correlated to the extracellular state?
No.
So what about all of those like hydrogen pumps and exchangers that we always talk about?
Perfect. So one of the reasons we talk about, and I didn't mention this, but on the cell surface, you know that the AMPA receptors have like a really interesting sort of topology that kind of looks like this, right? So the N terminal is out here, C terminal is out here, over here. Let me ask you, based on this and only this information, where is superecliptic fluorine? Where does it have to be? Based on what I told you in class, where is it? It has to be in the N terminal or somewhere that's sticking outside of the cell, right? Because that's the only place that's going to come in contact with the extracellular space. Here, this vesicle, in this vesicle, and remember when we talk about AMPA receptors trafficking, if you look back at some of the pictures, they're always trafficked in vesicles. They're removed by endocytosis in vesicles. Vesicles actually have a very low pH. When you look at the configuration of the receptor, it's actually inverted. So the N terminal is sticking up in here. As it fuses with the membrane, it gets into the correct orientation. So in these vesicles, the intracellular vesicles, the pH is low inside, and it's only when they're in the correct cell surface orientation that they're exposed to a neutral pH. Okay. Any other questions? Yes. Phosphate. What do you think they're removing phosphate from? I'm not trying to be obtuse, but no. Yes. So that's a good point. I made the assumption that you guys all knew that. That was a mistake. Okay. Yeah. So it's removing the phosphate groups on the AMPA receptors, because I made the assumption, and it was probably bad for me to have done this, that if you look at that picture, that when we have LTP, it's either due to more AMPA receptors, cell surface AMPA receptors, or the phosphorylation allows more to come in. So I made the assumption that if this is the case for LTP, LTD, if you remove this, there would be less. So that I made the assumption that you knew that it was on AMPA, and I have to apologize. It may have been inaccurate. Any other questions? Can you tell me which residue of, so you only have a few amino acids that can actually be phosphorylated. What are they? So tyrosine is one, serine is another. Where do you have a free OH group on amino acids? Threonine. So we only have these 3 amino acids, and it turns out that in AMPA receptors, it's usually the serine that gets phosphorylated that allows all these different changes to occur. Anyways, that's not stuff that's in your notes, so it's just for your own information. Other questions? Any other general questions? Like, you can ask anything, pretty much, because I think the stream is working, so everyone has equal access to it. Any questions? No? Still have time. Don't have any other questions. No? So how much confidence do you have in like your basic, like second year molecular biology? Pretty good?
Depends on what we're talking about.
Nice, okay. So when we talk about CRELOX, you guys are all comfortable with CRELOX systems? Because I know you learned it in second year.
Actually, we did it.
Yes, you did.
Excellent.
Bio 230.
Bio 230 did it. What?
Maybe Bio 130.
Nope.
CRELOX.
Everyone who learned it like that somehow came across it on their own. I
No.
promise you did. Yeah.
Okay. So, have you heard of CRELOX? Tomorrow you will hear about CRELOX. it will be a big part of like one of your questions on your final exam where you have to apply your knowledge. There is no set answer where you're going to have to tell me that you know how to design a construct and you're going to have to draw out the entire construct. So tomorrow we're going to be spending time going over the CRELOX systems that allow us to genetically manipulate cells. It's a very powerful technique that's used in neuroscience. You should all have a pretty sophisticated understanding of this. And tomorrow what we're going to learn is that the LOX-P sites, these are 34 base pair segments of DNA that we can design that bind specifically to this enzyme known as Cre recombinase. And as a recombinase enzyme, it will shape or remodel DNA. And it will only do it at these specific sites. So we can find a sequence, for example, and we can design these CRELOX sites in a number of different ways. So it's always 5' to 3', usually in DNA. Whatever sequence is here, you're going to have to understand like what's going to happen when you have these LOX-P sites, and what happens to the sequence when Cre recombinase is present. And you have to know that because starting tomorrow, we're actually going to look at a really elegant experiment that looked at if we were somehow able to erase a group of cells that originally held a memory. So imagine for a second that everything that you just learned today in this past hour, that somehow I was able to go into your brain and erase only those neurons that have kept all of this information. Pretty cool. And so what tomorrow you're going to learn is that there is a way to do this. There's a way to look at activated neurons and their synapses that hold that memory, and if we erase only that memory, then, or if we erase only those cells and their synapses, the memory also disappears. And the only way we can do this is using specific molecular techniques. So this technique here is going to be really important. So do you know about inversion versus excision? Have you heard of those terms before? So you invert a sequence of DNA or you remove a sequence of DNA. That's okay, right? So you guys all know about stop codons. What happens if you have a stop codon and you're in the 5' to 3'? Will it be transcribed or not? there's a stop codon. This is the sequence that I want. There's a stop codon before it. It won't read it, right? What if I could remove that stop codon? Would this sequence be read as long as it's in frame? So tomorrow we're going to learn how that can actually happen. Let me ask you this. This stop codon, is it specifically read in the 5' to 3' direction? So I need to have nucleotides in the precise order for it to work, right? What would happen if I inverted that sequence? It won't read the stop codon, right? So tomorrow we're going to learn about Cree locks. You're going to have to think a little bit about that. And I'm probably going to have to give you a lot of sort of extra questions so you can practice it on your own. I think the more practice and the more comfort you get with it, the better off you'll be in terms of drawing stuff out on the final exam, but you'll start encountering that tomorrow. So don't get worried about it, but I'm pretty sure I was told by students 2 years before. Okay.
You taught it in 360 though.
But I was told by students that took 360 the year before that you learned about this in Bio 230. They can't have dropped it in 1 year.
Maybe they have.
I don't think so.
Well,
Okay, I don't know.
I got a decent mark in Bio 230. I would have remembered learning it.
Maybe somebody erased your memory. Who knows? Do you have any other questions related to lecture 12? Yes.
Can you explain the thrombin and the tag again? The fluorescent tag with it?
There's no fluorescent tag in thrombin.
Oh, there is.
So I used an antibody tag or an HA sequence and then thrombin. But
The thrombin attaches to the HA tag, is that how it works?
what is thrombin?
It's a protein in the bloodstream, right?
Yes, what does it do?
It clots blood.
Potentially can. It works as a protease to activate different inactive larger molecules, like fibrinogen becomes fibrin, etc. So it's a protease, right?
Yeah.
And it's a calcium-dependent protease. So one of the things that, again, you tell me. So this is sort of this is the AMPA receptor subunit. So we know we have 4 of those. And let's say I put an HA tag on the N-terminal. Okay, so it's sticking outside of the cell. Where would I have to put thrombin to cleave that so that I don't see HA anymore? So would it help if I put thrombin over here? So I would probably put it here, right? So this is where I put the thrombin sequence. It doesn't interfere with the antibody to HA binding, because if it's there, it'll bind to it. But if I have thrombin here, thrombin will cut this, and everything that comes after it will be eliminated. I won't be able to see it. And so this means that I can't actually see an HA tag there anymore. So it's functionally like making it invisible. The receptor itself, the subunit is still there, the tag is missing. The antibody therefore can't recognize that. Do you have any other questions?
I think I understand the problem.
Okay, can I ask you a question? Okay, awesome. So if I did everything that we just talked about, so I had HA tagged AMPA receptor subunits on the cell surface, I applied thrombin to it, it worked, it cleaved everything off, and then I waited 15 minutes, and then I started to see HA tags again here. What happened? I can now detect them again. What happened?
So like the thrombin would be with the AMPA receptors, right? So if it resurfaces, I mean like the ones that didn't get cleaved inside the cell, once it surfaces.
So the most important thing was thrombin can't actually enter the cell. So as long as the cell is alive, there's no way for it to actually enter. So it only works on the ones that are out here. any receptor subunits that contain HA as well as thrombin down here at the bottom or inside the cell, they're protected. They can't be accessed by thrombin. And that's the reason why we see it. Again, I didn't go into any detail like how do you actually get them to start moving up to the cell surface. So normally we do this part where we inhibit the movement of receptors at 4 degrees. At 4 degrees, everything stops. Like literally, it's not enough to hurt the cell, but the movement of receptors stops. And then we cleave everything, so it becomes clean on the surface. And then at 37 degrees, when we put it back into normal conditions, then we start to see receptors moving again. So these receptors now start being inserted on the cell surface. Yes?
How do you kind of like stop thrombin that is there from the previous
You
experiment?
just wash it out. Just wash it out.
Huh?
You wash it out.
Okay.
So, so it's like taking, so we do this in cell culture. So in cell culture, like they're all sitting at the bottom of a dish. You throw in media that contains thrombin, it does its job. Then you remove that media and then you wash it in PBS, and that's enough to remove it. Yes. Any other questions? Yes.
Unrelated to lecture.
Okay.
When is our mark?
I don't know. I'm waiting for one TA to put in like the final marks onto the spreadsheet. So I have everything else ready to go. So I'm hoping as soon as possible. Why?
Because I wasn't know.
Yeah, I think a lot of students want to know. So I put up an announcement on the announcements page, and then sure enough, I got emails. When are the marks going to be posted? Where are they? So they're coming. They're coming. I'm not holding them back. They're just not all there. So you probably would have got your mark. No, N to Z, so no. M to Z, so no. You wouldn't have got yours.
Okay, I have to do this.
So all of your classmates would have got it except you, Tina. How's that?
That makes me really sad.
Yeah, that's why we didn't post them. Okay. Any other questions? So we're going to try to be either in this room or the room sort of downstairs, like every Tuesday. We'll do that. We'll see how the streams go and if they work. And yeah, if you have other questions, feel free to ask. It's a long sort of 4 weeks until your final exam, but they will go by really quickly. And the other suggestion that I have for you, if you can, go to the online library sort of repository, and the closest exam that would be beneficial to you would be the summer CJH version, because that final actually covers a lot of the similar material, and then parts of the April exam from 2017 as well. So those are the two I'd kind of focus on. And I will give you some other sort of worksheets to work on as well. If you are coming in to help with the study rubric, so I think Nicola, you said you might, tomorrow afternoon would probably work best. All right, try not to panic, it'll be okay.
Now that you say it, I feel much better.
No. All right, see you later. No, you don't. Nobody wants to panic. Oh, hello.